Hey everyone, welcome back to another video by Camroom 5. In today's video, we're going to be continuing the Biology O-Level ATP examination, which is now P4. And in today's class, we'll be talking about the question 2, specimen comparison. Most commonly in question 2, they give you two different specimens and they ask you to find out differences between how these two specimens look. So I like to follow a rule of sense. What does it mean? The first feature that you can look for is shape. You can comment on whether the shape is round, oblong, is it oval, is it cylindrical. These are various shapes that you can find. Is it curled or is it exactly straight? This is what you can comment on. Another thing that you can look for is additional features. For example, you can say that specimen 1 has more stains on it. Or if it's a cell, you can talk about which granules you see or organelles you see. You can talk about nucleus is visible or not. You can talk about cell wall is thick or thin. You can talk about the cell conditions like it's turgid, it's flaccid. You can talk about the orientation. These are also very important different features. Then you can comment on number. Okay, you can see more in specimen A or less in specimen B and vice versa. The fourth is size. People often confuse it with shape, which is not true. Size means is it larger comparatively or is it smaller, and that's it. This is what you do in size, but in shape you have multiple answers. Let me give you some examples from actual past paper questions. Let's begin with this. These questions are all from papers of 2015 till 2022. So these are not very old papers. They give you a very good idea of how to write answers. For example, let's see. You have to differentiate between cell A and cell B. You can see cell A, let me see. Cell A is right here. And if I'm not wrong, let me just make a circle around cell A. And sorry, this is cell B and this one is cell A. So you can clearly see that cell A is more rounded. So shape of cell A is rounded and B is not exactly round, it is more oval. So you can comment on oval. Another feature is, do you see any additional features? You see that cell A has more cellular organelles. So you can comment on cell organelles are visible. But you can see that in cell B nothing is visible. So you can say no cell contents are visible. You can also comment on another additional feature, which is their cell wall, you can say, because sizes are very similar, no one is bigger or smaller. So you can say it has thinner cell wall, which is another additional feature, and it has thicker cell wall. So this is how you comment on differences. Let's see this one. Here you are supposed to be commenting on the bone structure. So we can talk about that also. Let's see. Bone C, bone D. If you notice the size wise they are different or perhaps not. So you can say definitely bone C is smaller. So bone C is smaller. That's a very good start. And bone D is obviously longer, or you can say larger. You can also find some additional features or shape. So when you talk about shape, bone C is more curled, more bent. So it is more bent perhaps or curled. But when you look at bone D, it is more straighter. If you can look at additional features, you can try to notice additional features that in bone C and bone D, you can also find some features like bone C and bone D have different structures at the bottom. So bone C has a groove. You can see there's a groove over here. But in bone D, you can't see. So bone D is a little rounded from the bottom. Its base, its bottom isn't divided, but bone C has a groove. So there's a groove at bottom of bone C, and it's more rounded without groove at the bottom. This is how you can write differences. Let's see this one. Here you can also find various leaf structures and you have to find differences over here. So they have already given you that shape and edge are the features and they're concerned with laurel and oak leaves, if I'm not wrong. So when you look at laurel, and they want you to comment on the shape. So the shape is more or less oval with pointed at the top. So you can say oval plus pointed at top. You can say that, it's totally okay. So you're commenting on the shape. When you're concerned with the shape of the oak leaf, you can also say that oak has a very irregular leaf structure with leaflets coming out of it. So irregular structure is there at the end of the day, and you can say leaflets are here. People who compare it with the word we use in liver, you can say lobes are coming out of it. So this is how you talk. When you comment on the edge or the margins, so you can also see that when you talk about laurel, let me show you, it has a very smooth kind of edge. You don't see much points coming out of it. But when you talk about let me see, no, yeah, even oak has a very smooth edge. So both have smooth edges. I won't say that one has unsmooth edge, because even oak leaf has a very smooth. This is not smooth. This is not smooth. Which does go with the fact that its name starts with the letter horn. But anyway, coming back. So let's do this one. Complete the table to identify two differences between the two pictomicrographs. And if I'm not wrong, these are alveoli structures. So you can find various differences over here. First, let's talk about the smoker, and let's recall that we can talk about sizes. So the alveoli, you have to concern yourself with the alveoli and the overall structure. So the smoker has bigger alveoli, and there are many alveoli, so they are larger alveoli. You can just dedicate one point to larger, because that is a reference to size. So larger alveoli, and you can say smaller alveoli in the non-smoker. Another thing you can see is there are additional features possible. So let's look for additional features. Let me magnify it even more. The walls are broken in the smoker's alveoli, but the walls are more structured in the non-smoker alveoli. When you talk about the smoker, you can see that there are fewer blood vessels. I can see there's a blood vessel over here, but no, that's not. Okay, this is definitely a blood vessel. This is a blood vessel. But when you look over here, this is a blood vessel. Over here, this is a blood vessel. This is a blood vessel. These are veins. This is a blood vessel. So you can identify various blood vessels over here. So perhaps in smoker, you can say there are smaller or fewer blood vessels. And here you can say there are more blood vessels available. So this one was indeed tricky. I have one more question for you guys. You can see figure 2.1 shows red blood cells from a frog, and figure 2.2 shows red blood cells from a human as seen under the microscope. You want to comment on shape first. So shape wise, when you notice the overall shape, you can clearly see that the shape of the frog's red blood cells is more oval, but the human blood cells are more round. Overall, you can see they are round. You can't comment on the 3D shape because this is not very clear. They are more thinner from the center. Is that what you can say? But shape is a very simple word, and you can call it round. When you talk about nucleus, so you can clearly see that the nucleus is visible here and nucleus is absent here. So that's an additional feature. So here you can see how we did differentiation between various structures. In the next video, we'll talk about more stuff from P4 of bio. Stay tuned, guys. Thanks.